Good morning. Thank you, Michael. Thank you, Klaus, for inviting me to present here. I was also at the very first SLTBR meeting, not at the founding meeting, which I think Bud, Norm, and Michael were at, but the first annual meeting held 27 years ago in Bethesda. It was very meaningful to be part of an organization where, as a first-year fellow, one can do research, work with world-class researchers, and then present those data in a forum like this and get feedback from across the whole world. So it's very meaningful for me to be here again at an SLTBR meeting. Michael asked me to speak on the topic of the biology of circadian rhythms, and so I'll begin with a philosophical point. With regard to this, some of you, I'm sure, are more expert in this topic than I am, but I really intend to present this as at an overview level because this is a teaching course. Now, again, the instructions to me were to think in terms of the biology of circadian rhythms, but from a philosophical background, I'll read you a quote from Ernest Rutherford, the father of nuclear physics. What he wrote was, "All science is either physics or stamp collecting." Sadly, I'm not a physicist, and this isn't the right forum to go over physics, but I think to the extent that we as researchers keep the physics in the back of our mind, the more it will have to inform us. But as I said, I'm not a physicist, so I'll be presenting to you confessions of a stamp collector, which I am. In particular, this is a stamp of from Austria showing a clock. And from a high-level point of view, and some of the slides will get into the weeds, but I would encourage you not for this purpose to get into the weeds, but to think about the biological clock really has a lot in parallel with a traditional clock. Now, again, most of us don't use wind-up clocks like this anymore, but I think it's illustrative because there are analogous physical aspects and structures to the old-fashioned wind-up clock. For example, there is output, and let's see, not working so well. The output on the clock is in the hands of the clock, whether it's showing hours or minutes or seconds, and there are ways that our biological clocks have an output and demonstrate things. There's an input to the biological clock, and in fact, the more that scientists are learning about it, just like there is one dial to set on this analog clock, there's a dial to set what time the alarm goes off, to set what time the clock thinks it should be at any certain time. There's also inputs to give energy to the clock and to give energy to the alarm, and in fact, these are part of the human and animal clock as well. The inside is not at all simple. Just like in an analog clock, there's all these different parts and gears, and it's certainly even more so inside the biological clock. Nonetheless, there are some general principles that we can go over. So, this biological clock exists in our brains, and this is a sculpture by Gerstein called The Last Moment, and indeed there is a representation, in fact many representations of the outside clock within each one of us. Why does this occur? Well, if we think back to the beginning of time, since the very beginning, every creature, every species of life, at least on the surface of our planet, has been circling the sun on an annual basis. The moon circles the Earth on a 30-day basis, and the axis of the Earth is spinning around on a daily basis. And the sun, being the central source of light and the central source of energy to this planet, is something that has provided a stimulus to every species of life on the surface of the planet. And I'd love to show this slide from Arthur Galston's book on biological clocks. This shows a picture of the Datura, commonly called the angel's trumpet flower. And the angel's trumpet flower has a biological clock so that every night, if you will, it goes to sleep, it closes its petals, and at dawn the petals start to open, they get wider, and full bloom during the whole day. And of course, as plant scientists began to discover hundreds of years ago, this is regulated not just by the sun, which sets the time, but it's also regulated by an internal biological clock. Put the plants in darkness, and these plants will continue to for at least several days to continue on the same schedule of gradually waking up each morning, opening their petals to the sun, even if it's not there, and then closing them up. And I love this slide because I would argue that it's this biological clock, the sensitivity to light in terms of changing the timing of the biological clock, the waking, the sleeping. This it could conceivably in some ways be the most fundamental form of behavior conserved across every species of life on the surface of the planet in different ways. Because all of us, when we psychiatrists and psychologists talk about behavior, we have many different kinds of complex terms. Freud wrote books and books about them, but the core one, I would argue, is this fundamental biological clock that in some ways wakes us up and puts us to sleep every day. And indeed, the biological clock affects not just plants, but it affects all kinds of animal species, and we humans as well. Now, there are some caveats for this presentation today. The first one, I write TBD, to be determined, and that what I mean by that, there are literally thousands and thousands of details, more being discovered each week, and as science could and should do, some of the things that we thought were true 20 years ago, we learned 10 years ago, are not true, and some of the things we think are true today, over the next couple of years, will surely be disproven. The next point, things are not so simple. It's very, for teaching purposes, it's the job of professors to simplify things. The reality we are always finding is more complicated. The other caveat for today is that most of what I'm going to be talking about I'm going to present in terms of what is known about mammalian species. There are a lot of commonalities with non-mammalian animals, but I'm going to focus more on mammals, including we humans. So this is the weeds. Don't get scared by it, but what I want to emphasize is the three different parts of the biological clock that are known and operate within humans. First of all, there is the input. The primary input to the biological clock is light. Again, this is not the only input, but light is the strongest input. Light, especially for purposes of resetting the biological clock, of a blue light, 460 to 480 nanometers, enters through the eye. And again, we'll come back to this. This is an oversimplification. The light from the eye sends signals through the neurological tract to the internal part of the major core of the biological clock, the suprachiasmatic nuclei, which is in the hypothalamus. I'll say more about this later. Then neurological signals are sent from the SCN to the paraventricular nuclei, down the spinal cord, and we'll come back to this, and signals eventually go up the spinal cord to where the body starts to generate the major output that we can detect. So we don't have hands of a clock, but we have production of the famous hormone melatonin, which is largely, not exclusively, but largely made in the pineal gland and then circulates into the bloodstream. The melatonin that is produced from the pineal gland both travels through the body and gives signals to the rest of the body. It also goes back through the bloodstream back to the suprachiasmatic nucleus where there are melatonin receptors and can reset the clock itself. But we'll come back to more of that later. From an anatomic point of view, let's look at some of the major players with regard to the brain. So we have, the eyes are not shown in the brain, but the eyes send a neurological pathway through the optic chiasm. This is where the left eye and the right eye optic nerves meet, and then send tracts to the suprachiasmatic nucleus, which is a set of nuclei right on top of the optic chiasm in the hypothalamus of the brain. Suprachiasmatic nucleus, and I really prefer to call it nuclei because there are two of them, and it's really a collection of thousands of individual cells, is the master clock for the body. And again, the signals go down and up the spinal cord, and eventually, with regard to the melatonin output, the melatonin is largely produced in the pineal gland, which circulates melatonin through the bloodstream to the rest of the body. Now, one particular reason why I appreciate being able to give this a teaching talk today is because it does give me a chance to pay homage to three of my mentors who I learned a lot about, great scientists and great human beings who are part of this organization. So I'll mention their names in particular. So this slide was adapted from a paper that Tom Ware and Norm Rosenthal, sitting here in the second row, who I had the great privilege to work with at NIH, prepared with regard to seasonal affective disorder. And this shows that pathway that I talked about previously. Again, the sun being the primary input to the biological clock sends its signal of light to the eyes, especially to the retina of the eye. You see the neurologic pathway going to the SCN, the suprachiasmatic nuclei, then to the paraventricular nucleus, and again, this is oversimplified, down the spinal cord to the intermediolateral nucleus, back up to the superior cervical ganglion, and then through a neurologic pathway to the pineal gland. So this gives a rough outline, or actually a detailed outline, of the key neurological pathways. Now, the SCN, the suprachiasmatic nuclei, play a critical role in terms of synchronizing peripheral clocks. What I mean by that is that the more people have looked for biological clocks in the body, the more that they've found them all over the body. In fact, you could say that our bodies really in many ways are a collection of biological clocks, with the SCN, the suprachiasmatic nuclei, being the master clock. But at the same time, there are other clocks. For example, the thyroid gland, you can see circadian rhythms, circadian being 24-hour rhythms of different hormone production from the thyroid gland. The SCN sends signals, at least via melatonin, to myotubules in the skeletal muscle, again, 24-hour rhythms. The pancreas is regulated with the biological clock. Again, you can see 24-hour rhythms. These diminish over time because this is in a model where the light was taken away, so just the internal clock stays true for several days before it stops cycling without the light structure, the light stimulus. Skin fibroblasts as well, and this is really just a starting list, but these are all kinds of output. Now, one key point to make is that the connections between the suprachiasmatic nucleus and the rest of the body, these signals of timing are both through humoral, in other words, through blood chemical components, and through neuronal pathways. Now, the SCN, the suprachiasmatic nuclei, are really fascinating. One of the ways that we know that they have convey humoral outputs is that this is based on a study by Toussaint and Meisel, published about 10 years ago, where they established in mice that cyclic information from the hypothalamus was lost after the SCN was ablated. So let me show you what's going on in this slide. So this is a stylized suprachiasmatic nucleus, and you can see it has a 24-hour rhythm of output, both electrical activity and in chemical production. When the suprachiasmatic nucleus is removed, is ablated, the output, the 24-hour output, circadian output from the SCN is lost. But when you take a graft in these mice of an SCN, put it back in, the 24-hour rhythms could be restored. Now, what work published by Silver et al. Did was that they took these grafts and encapsulated them, so preventing neural outgrowth, and they were able to show that by a non-neural connection, not by nerves, but by humoral factors released from the SCN, 24-hour rhythms are still able to be produced in the body. So it's not so different from other aspects of human physiology where there are sometimes multiple parallel or redundant pathways for similar purposes. There are other factors also that regulate the peripheral clock, so how much food we eat and what time we eat, our locomotor rhythms, what time we're waking up, with what we're doing exercise, can affect biological clocks body-wide. Let me go on now to the inner mechanisms. specifically of the superior chiasmatic nucleus, the suprachiasmatic nucleus. Now, this is an oversimplified slide. It was once presented as truth, not by me, but by the people who presented it. But again, truth becomes more complicated as we learn more about it. Traditionally, the suprachiasmatic nucleus is considered to have two subdivisions to it. A dorsomedial section, a shell, and a core or ventrolateral section. To a large degree, the dorsomedial section receives non-photic, non-light input. This is places where the melatonin receptors are. The ventrolateral, situated right next to the optic chiasm, has neurons that receive light-activated input. So this is where phase shifting, changing of timing of the biological clock takes place. These SCN is composed of literally thousands of independent cellular oscillators that are coupled into a neural network. One of the, I think, neat findings that's been discovered in the last couple of years, and this is a slide from Buhr and Van Gelder from 2 years ago, was that they were able to establish how the SCN regulates itself. Because remember, I mentioned that it's composed of thousands of individual clocks. Well, how do those individual clocks get on the same page? And it turns out that we have a peptide called LHX1, and it is produced in the SCN, and it produces coupling peptides to make circadian rhythms. And if the production of LHX1 is stopped, the circadian rhythm is damped, and the SCN's consistent rhythms are limited. So this provides very good evidence, and because LHX1 is unique to the, at least thought today to be unique to the SCN, this is thought to be what helps make this clock different from all other clocks in the body. And it turns out that light that can cause phase shifts, and we'll be talking more about this morning. In other words, light that's able to change the time of the body clock has a specific effect of suppressing the production of LHX1. So it's really a rather fascinating system. When light that can cause a phase shift is shined on the organism, the LHX1 production in the SCN is suppressed, which makes for a less coordinated SCN and allows the SCN to shift more easily, to cause a phase shift. Now, within the SCN, GABA is the most common neurotransmitter throughout. These two nuclei, one on each side of the optic chiasm, separated by the third ventricle, communicate both by synaptic junctions and by release of vasointestinal polypeptide. Now, the SCN is thought to have a major role as being an irradiance detector. And it gets its input, and we'll come back to this, from different signals that are together added up or integrated from a molecule in the eye called melanopsin from rods and cones. The SCN has cells that are acutely stimulated by light, and it also has cells that are acutely inhibited or silenced by light. In other words, there are some cells that are turned on by light, some cells that are turned off by external light. And one of the, again, findings from the last couple of years of the literature that I've found most interesting is one question that I've always wondered is, what is it biologically that makes a daytime animal, a diurnal active animal, different from a nocturnal animal? And I'm sure more and more will be learned, but one of the interesting findings is that daytime active animals are thought to have relatively more cells that are turned off by light. So this is, I think, a very important biological finding. Now, light activation and light inhibition of SCN cells is a function of light intensity, how bright the light is. It's a sigmoidal relationship with a working range specifically centered around the light intensities of dawn and dusk. In other words, the SCN, the suprachiasmatic nuclei, is particularly a day versus night detector, and it's very sensitive, and we'll come back to this, specifically to the light levels that occur at around the time of dawn and around dusk. So again, Michael Terman's done tremendous work with Dawn Simulator, and that's maybe one of the reasons why this is an effective treatment, because the signal to the SCN is particularly sensitive in the SCN at times of dawn and dusk. Interestingly also, the SCN is only really sensitive to light-induced phase shifting during the dark phase. So in the middle of the daytime, and we'll hear more about this morning, the SCN is not really sensitive to changes in the biological clock. Behavioral activity correlates with electrical activity in the SCN. This is mouse data from Hoban et al. From 7 years ago. The SCN also retains a memory for photoperiod. What does that mean? It means that, again, as I talked about the plants, that if you turn off the light, the plants will continue to wake up and go to sleep every day for several days. If you put someone in a cave, we will continue to wake up and go to sleep at similar times for at least several days because we have a memory. And similarly, that memory is kept in the suprachiasmatic nuclei especially. We're going to hear, I'm sure, a lot more this morning about the PRC, the phase response curve to light. This graph, it's a complicated graph, but the key thing to show for now, and you'll see more later, is that, we have, during the daytime, the effect of light on changing the time of our biological clock is relatively little. But during the dark hours of the night, in the first half of the night, shining light, which gives a signal to the SCN, causes what's called a phase delay, where we wake up later. In the latter part of the night, bright light applied to the person, to the eyes, causes a phase advance. So what's going on inside the SCN that allows these things to happen? So this is sort of the inside part. Essentially, and again, this is available in most common textbooks. This is adapted from Reiter et al., 2010. I think this will be, although you're not supposed to take pictures and videos of this, this will go on the web through New York State Psychiatric Institute, so you'll have access to this. But basically, the signal from the eye tells the SCN to synthesize genes and I'll share a bit of that in a moment, leading to the signal going to the paraventricular nuclei, to the intermediolateral cell column. Now, the genetic story inside the SCN, I could talk about, or someone who's truly expert in it could spend a day talking about it and not be half done. So what I want to share for the purpose of this teaching course is a really simplified molecular model of the circadian oscillator in the SCN. So essentially we have a 24-hour period, a 24-hour clock within us, and we continually have production of transcription factors, in particular clock and BMAL1. As the levels of these transcription factors go up, they lead to the transcription of genes that are controlled by the biological clock. Some of the more prominent ones are PER1, PER2, CRY1, CRY2. When you get extra high levels of these, you trigger a negative feedback loop, which then reduces the production of clock and BMAL1 so that you have a continual rise and fall. And again, this is far simplified, but it gives you a sense of the basic pattern for how the clock is working within the SCN. In what I consider to be really a fascinating finding, a mutation in the PER2 gene has already been associated with familial advanced sleep phase syndrome. So this is familial advanced sleep phase syndrome is a disorder, a genetic disorder within families where people typically wake up several hours early compared to the rest of the world each day, go to sleep several hours early, and it's not within their control. It's not a voluntary phenomenon. It is genetically driven. And the fact that today we can link, at least in some families with this, this disturbance to a genetic abnormality in the biological clock is really a beautiful work in science, and I think a a great model for all of us who are trying to connect clinical phenomena and clinical disorders to the biological clock. Certainly the literature is, one of the reasons for our field and for this organization has been to try and to unleash the connection between the clinical disorders and the biological clock, but it's not always so easy to do that. So again, this graph from the Ware and Rosenthal paper, I've gone over it before, but it relays again the pathway. And this is something from a take-home point of view, it is worth having a general understanding of this pathway from the eye to the suprachiasmatic nucleus and the hypothalamus to the spinal cord and back to the pineal gland as far as the inner workings of the biological clock. Now, in the pineal gland, one of the major jobs of the pineal gland is melatonin synthesis. Fascinatingly, melatonin is a hormone of the dark. Whether you're talking about a bat that's active at night or a human being that's active at day, all mammals, all animals that I know of produce melatonin specifically at night. So it's not a marker specifically of behavior, but it is an internal marker of the external environment. For today's purposes, we don't have to go through, you can do this at your leisure if you're so motivated, the exact chemical pathway within the pineal gland that leads from the norepinephrine signal to the gland, to the production from tryptophan and into serotonin, into melatonin, which is then released into the capillaries. The melatonin released into the capillaries, it can go back to the suprachiasmatic nucleus through the ventricle in a negative feedback loop. There are melatonin 1 and melatonin receptors throughout the body. Melatonin is a very lipophilic molecule. That means it's a very fatty molecule, so it can penetrate inside the cell nucleus, potentially of every cell in the body, and have additional effects, which remain to be proven what they specifically are throughout the body. But there are well-described melatonin and melatonin 1 and 2 receptors. Not Michael Terman, but that's a good way to remember it. You can see that nocturnally, some animals tend to produce melatonin more during the long nights of winter, of shorter melatonin production during the short nights of summer. The melatonin 2 receptor is argued to be sleep promoting. It's not within the scope of this talk, but there's extensive debate and discussion in the literature about, is melatonin a sleep promoter? Is melatonin a good sleep medicine or not? I will throw in my 2 cents here just as a way to rouse people, and then if anyone wants to comment on it, they can do that later. My own personal reading of the literature is that melatonin can, at least immediate release melatonin, the kind that the body makes, can promote sleep, but it promotes it at times of day when we don't have circulating melatonin. So if you wanted to take a midday nap, it might be a very useful tool. The evidence showing that its efficacy is good at nighttime is very limited and extremely debated. It's a highly controversial issue. There are slow-release forms of melatonin which may in fact be very useful for bedtime, for sleep as well. All right, let's say a little bit about the output. I mentioned the peripheral rhythms in the thyroid gland, the skeletal muscle, the endocrine pancreas, skin fibroblasts. But in addition, melatonin and the biological clock output shows up in brain activity, heart rate, blood pressure, renal function, general metabolism, immune, feeding, body temperature. I mean, it seems to be the more that people look for circadian rhythms in different aspects of our biology, the more that people are finding them. I'm going to finish by talking about the input to the biological clock. So again, here, this shows, especially with regard to phase shifting, changing the time of the biological clock, light of blue visible light acting on melanopsin through the retinal ganglion cell. These are critical to clock setting in the suprachiasmatic nuclei. But rods and cones are also thought to provide input as well. So it's, again, the story, the more we learn about it, it's more complicated than it's first stated. First it was stated that rods and cones were responsible for it. Then it was stated that melanopsin is responsible for it. Now it seems that there are multiple critical inputs. And again, our behavior response to light is so complex that we may do ourselves a disservice if we just attribute all of our behavior to one or two molecules. Now, melanopsin, it's an active area of research. There are at least two critically important forms of melanopsin, one called 11-cis-retinal melanopsin. This is the structure of the chromophore of melanopsin. Chromophore is a word that refers to the part of a molecule that absorbs light. And you can tell because it has all these pi bonds, alternating double bond, single bond, double bond, single bond. Throughout biology, wherever you see a molecule like this, it tells you that this molecule is capable of absorbing light. It doesn't tell you that it's physiologically important, but it tells you that it has that capacity. And there are two what are called bistable forms of melanopsin, the cis-retinal and then trans-retinal. And basically light of about 467 nanometers causes this part to flip up to become the trans form. 476 flips back, causes the molecule to flip back like this. What the specific role of each of those two different wavelengths is, as far as our biology, remains to be determined. There may also be at least a third form of melanopsin that may play a critical role as well. With regard to the eye, this is a slide of a monkey retina, and if you imagine that the source of light is at the top, this is the front of the eye, this is the back of the eye. Light comes in through the front, goes through several different layers. Near the front is the ganglion cell layer, where the retinohypothalamic tract photosensitive ganglion retinal cells, these are the melanopsin-containing cells, make up about 1% of the cells in these layers. Light goes through to the rods and the cones, and you can see lots of blood vessels at the back of the eye as well. Now, work done by Walmsley et al., published in PLOS Biology last year, shows that the signal to the eye, it's not just a matter of brightness, but it's also a matter of color detection. It turns out, we have different kinds of rods and cones specifically in the eye that are more sensitive to one color than the other. And because the change in the amount of yellow versus blue light is particularly important at times of dawn and dusk, it turns out that our eyes are not just detecting the increase in light, but the change in color, and that provides a clear signal to the SCN that the body is waking up. This is work I've been able to praise Michael, I've been able to praise Norm Rosenthal. This is work by Gooley et al., including Bud Brainerd, who a giant in the field, also part of one of the founders of this group. This work that they published a few years back has shown that there's more than melanopsin, however, that meets the light in the eye. They did work where they looked at two specific wavelengths of light, a blue wavelength and a narrow green wavelength, and they found that both the blue and the green lights were equally effective in turning off melatonin, because that one thing that light does, and this is work shown by Al Louie, in humans, light can turn off production entirely of melatonin in the body. And what Gooley et al. Found was that the effect of green light, it's strong initially, although it decays over time. But they found that the time-shifting effect of the green light cannot be explained by melanopsin alone. And indeed, we learn about more and more molecules. The work of Ramey, Whereas Justice, Juan and Michael Terman, Tassini and Menaker led us 20 to 30 years ago to see evidence of an autonomous biological clock, a light-sensitive clock in the retina, even independent of the SCN. And one of the things that's just been discovered, published in papers in the last couple of years, is that the biological clock in the eye can be entrained to light even in the absence of rods, in the absence of cones, and in the absence of melanopsin. In the eye, the retinal entrainment was most sensitive to ultraviolet A and violet light. And work done by Buer and Van Gelder has shown that another molecule, another light-sensitive molecule called neuropsin, in the retina ganglion cells, that may be this particular molecule required to be the photosensitive molecule to mediate these effects in the eye. I'll just take 2 minutes to look further beyond into my own particular interest of something that I've called humoral phototransduction, the idea that there's a conservation of circadian and seasonal responses between plants and animals. It grows out of the idea that plant chlorophyll its chromophore is virtually identical to the light-sensitive part of human hemoglobin. And indeed, this is not just a chemical coincidence. They're in fact plants and mammals form these molecules along the same biological pathway, starting out with delta-aminolevulinic acid as a precursor. Plants get magnesium added to the center and go on to become chlorophyll. Animals get iron added and go on to make heme. Also, this is beyond the scope of today, but the heme in plants and animals gets converted to biliverdin, and then plants use that to make phytochrome, which is perhaps the most important time-setting molecule in the plant. We use it to make bilirubin, which, at least when I was in medical school, we were taught was a waste leftover product of evolution. It turns out that light causes the dissociation of carbon monoxide and nitric oxide from the heme of hemoglobin. And one of the things that I find fascinating, I'm putting up again the slide from Gooley et al., where they showed the importance of blue and green light. I should say that there are, what color light works best for SAD is a source of great discussion. There are some papers arguing blue, some arguing green. Certainly no one can argue with the usefulness of white. But one of the fascinating things is that how is this green light having an effect? And at least in photobiology, one common understanding or one way of doing the research is to be able to link a particular biochemical effect to a particular wavelength. And it turns out that the peak wavelength for this process of causing heme to release CO, that peak effect occurs at 540 to 570 nanometers, right in the same region as the green. So whether this study is proving that cones are having this effect, or could it be hemoglobin CO dissociation, I'm not sure, but it's something to think about. And the proposal of this model is, in fact, that gas neurotransmitters are produced in the eye, go back through the retinal vessels to the cavernous sinus. And I should say the cavernous sinus is neither cavernous nor is it a sinus. It's a network of veins that wrap very tightly around the internal carotid artery, which could easily allow for transport of gases like CO and NO directly to the suprachiasmatic nucleus, which is right there. And in fact, in the 20 years since this model has been proposed, it has already been proven that the following chemicals, GnRH, ACTH, LH, da-da-da-da-da-da-da, all cross through countercurrent transport from the cavernous sinus into the internal carotid arteries at this place. Some drugs even do that as well. I could speculate that intranasal ketamine may be having some of its effects via this route as well. Data that supports this model include the work of Kozierowski et al., published 4 years ago, where they showed that in a pig model, in a seasonal hybrid pig model, they showed that there were elevated levels of CO in the eye blood versus the veins in the daytime, not at night, only in summer, no difference in the winter. And in a plug for you to come to my poster tomorrow, we have some preliminary data showing in the pig model, venous CO increases following 5,000 lux light exposure. And as I see Michael standing there, I see I'm out of time. So, any questions? Norm.
I'm going to do something very obnoxious, and that is that people ask any questions and somebody stands up and makes a comment and there's no question. So, in any event, I would just like to firstly compliment you on your scholarly and very lucid presentation, and to say that one theme that came out was the theme that when you've got biological function that's as important. Thank you. As circadian rhythms, when something is as important as circadian rhythms, there's not going to be one pathway that is going to be responsible for orchestrating such an important function. and you've shown this obviously in many different aspects. I, in turn, working in a clinical research situation, have similarly seen that. For example, Tom Ware and I don't know if you were there at the time, but we did an eye versus skin.
That was before my time.
Study where we looked at whether the effects of the antidepressant effects on seasonal affective disorder were mediated by the eye versus the skin, and the eyes won in that particular experiment. However, there is now extensive evidence that UV light on the skin actually has euphoriant effects. They're suggesting that it may be beta endorphin related through melanocytes, and certainly tanning salons anecdotally and experimentally have been found to be beneficial, although of course the cost of the skin cancer risk is not worth it. So there you get redundancy in skin versus eye. A conversation that Martin and I were having yesterday reminded me of a patient I had, blind, who had had her both eyes enucleated because of cancers in the eyes, who retained a very clear, and this was done very early in life, retained a very clear seasonal affective disorder pattern. So whether that was coming through light or whether it was coming through some non-light mechanism, I don't know. Thank you.
I can't argue with anything you said.
We have a few minutes for questions, debates, clarifications. Yeah.
Yeah, I'm wondering, hi, my name is Stefan, I'm a PhD student, and I'm wondering, as a student, I didn't read the paper by a writer, but why is the pathway going all the way down the spinal cord and going back up? Is there a reason for feedback or something, or?
Yeah, I've asked that question myself every time I see that slide. You know, if one is looking, I mean, I think it's natural and it's appropriate to look for evolutionary, teleological kinds of explanations. I don't have the answer. Someone else may. No, but it's a very interesting question, I think, and really would be worth pursuing. Actually, on the subject of questions, Norm, you're bringing up the eye versus skin study. Let me make one plug in that was one of the studies that got me so interested in this field, because there was a very clear hypothesis with a very clear. The study had a very clear hypothesis, and a clear result was being explored. And so often in the work that we do, we are just gathering data. We're stamp collecting, if you will. And to the extent that we can have precise hypotheses to try and show something true or false, whatever the outcome, I think it advances the field. So.